Good afternoon. Welcome to Denver Museum of Nature and Science Facebook Live event. My name is Paula Cushing. I am one of the curators at the Denver Museum of Nature and Science. That means I'm one of the scientists behind the scenes at the museum. And my research specialty is arachnids. I study spiders and their relatives. But today I'm going to be talking about something that is also near and dear to my heart. I consider myself a taxonomist and systematist. I study the diversity of life on Earth. And what I want to talk about today is naming new species and discovering new species and how that process happens. So we'll talk a little bit about species discovery, how that happens, and then what goes into naming a new species. So one of the reasons that I do what I do, if I can move my slides, is because I'm fascinated with the diversity of life on Earth. And all of the curators, all of the scientists who work behind the scenes at the museum, one of the things we all have in common is we are all fascinated with diversity and with how that diversity on this planet evolved. So whether you're talking about space scientists, they're interested in understanding the processes by which the diversity of the universe formed and the processes by which this one planet in our solar system formed that allowed and enabled life to evolve on this planet. My colleagues in anthropology are interested in cultural diversity and the diversity of humans throughout the world. My colleagues in earth sciences and paleontology are interested in studying past life on Earth, the extinct life that is no longer with us. And those of us in the zoology department are interested in the current life forms, the current species that have evolved on this planet. And when we talk about Earth's diversity, what's fascinating to me is that we have only touch the surface in terms of knowing what other species we share this planet with. So this, by this one estimate, these scientists estimated that the true species diversity on Earth, the number of species, is probably between 8 and 9 million species. And yet we have only discovered and described about 1.2 million species. So if you think about that, that means in the same paper, they estimated that 86% of the species that exist in terrestrial habitats and 91% of species that exist in marine environments are yet to be described, are yet to be discovered. That's kind of mind-blowing if you think about it. So let's talk about how we document that diversity. And natural history museums like the Denver Museum of Nature and Science play a big role in documenting that diversity. Behind the scenes, we have collections of specimens that represent the diversity of species that are found on Earth. And what does that discovery involve? Well, it literally involves discovery. It involves going out into the field, going out into nature, going to different environments, doing field work, and making discoveries of specimens that oftentimes you look at and you think, I've never seen that before. I think this represents a new specimen, a specimen that represents a new species. So once you've made that discovery, then what you do is you bring it back into the lab. You compare those specimens to existing specimens that exist in museum collections. You compare your specimens using microscopes or using other means. You compare the mystery specimens to existing specimens that represent known species to convince yourself that, yeah, I really think this is something new. You look at the morphology, you examine the morphological characteristics, the anatomical traits, to see if they are different than existing species that are already known to science. You then compare it to the literature. You go into the primary literature, the species descriptions that have already been written, and we'll talk more about how those are written. and you compare your mystery specimens with the literature. So you compare them to real specimens, you compare them to illustrations of existing specimens that have already been described. You might even use DNA analysis these days to see if the genetic data, if the DNA sequences, this is an example of a DNA sequence, is the DNA sequence, does it suggest that your mystery specimens represent something new? You might even run your molecular data and your morphological data and create an evolutionary tree called a cladogram or a phylogeny. And this is another line of evidence that what you've collected may represent a new species. And if by doing all of that, you convince yourself that yes, in fact, you have collected something that is new to science, a new species, then you go through the process of publication. And that's a very involved process. So this is an example of a monograph, a huge paper that described one genus of tarantula-like spiders in the genus Eumidia. This paper just was published in 2021 by my colleagues Rebecca Godwin and Jason Bond. And in this paper, they described over 30 new species in this genus Eumidia. And they recognized they were new species by going through that process of discovery and comparison with the existing specimens and determined that yes, and they used DNA analysis to determine that yes, these 30 plus species are new to science. So then in this paper, they then describe the new species. And the species descriptions involve, one of the fun parts is involving the people who write the species description get to name the new species. So they get to come up with a new species name. And I'll show you later in this lecture some of the fun species names that are out there. But in this case, they name this new species Eumidia rongodwini, after the first author's husband. And that is described in the etymology, which explains the derivation of the new species name. But let's look closely at that new species name, because the species name involves the first part of that species name is the genus Eumidia. The second part of the species name is called the specific epithet. So the combination of the genus and the specific epithet together, those two parts together, that makes up the name of the species. And typically when you see the name of the species in publications, it's followed by the describer's name. So in publications from henceforth, this would be called Eumidia rongodwini, Godwin and Bond, because Godwin and Bond are the ones who described it. In the species description, you see a description of where the type specimen was collected. And let's talk a little bit more about what that means, what the holotype is. So when a scientist, particularly in animal sciences, is describing a new species, they pick one specimen. Could be a male, could be a female. If it's a botanist, they'll pick one individual plant specimen. But that individual, upon which the species description is based, is known as the holotype, or abbreviated to type. But the holotype is the most important specimen that exists. And these days, it is required that type specimens be deposited in museum collections. That type specimen is the specimen upon which the entire species description is based. And then further into the species description, the describers will capture diversity, morphological variations, by examining other specimens that are called specimens in the type series. But the holotype, that's one specimen. So this paper describes the type locality, where they collected the type specimen of Eumidia rongodwini. Etymology explains that the name is derived from the name of Ron Godwin, the husband of the first author. So it's an honorific, it's in honor of Ron for the support that he gave to the first author. Diagnosis section explains why the researchers felt that this was different than any other species in the genus Eumidia. So the diagnosis explains the differences that distinguish Eumidia rongodwini from all other species of Eumidia. And the description of the male holotype will then go into detail about measurements, how big the specimen is, what its colors are, any other morphological characters that are involved in that distinguish it. And then there's more to the species description, but these are the key parts of a species description. So this is a fun little factoid. Guess what one species on Earth does not have a holotype designated for it, a type specimen? You're right, it's us. Homo sapiens does not have a type specimen. It's interesting, isn't it? So from here on, I just want to show you the names, some fun names of species that are out there. So my colleague Peter Yeager discovered a new species of spider in the genus Heteropoda, and Peter thought that the crazy hairdo of this spider reminded him of one of his favorite artists, David Bowie. So he named it Heteropoda David Bowie. This researcher discovered a new species of louse, and this researcher Clayton was a fan of the cartoon, the Far Side cartoons by Gary Larson. So he named this louse species Strigophilus garylarsoni. This species, this genus of spiders right here, my colleague, also Peter Yeager, wanted to honor Greta Thunberg, who is the young climate activist. So this entire genus is named Thunberga genus after Greta Thunberg. I've been fortunate enough that I've had a few species named after me. In the same paper that I mentioned, they named a species of this Eumidia genus, Eumidia polycushingi, after myself for the work that I've done in the field of arachnology. So that was a real honor for me to be included in their paper and to have an honorific, have a species named after me. Several years ago, I also had a fossil sulfugid named after me, and the genus itself is named Cushingii genus. And this is because of all the research that I've done with sulfugids. And then that same year, several years ago, a colleague produced a paper where he described new species of ant-mimicking spiders, and he named one Laura Cushingii after myself, because of work that I've done with spider-ant associations and ant-mimicking spiders. So that's a real honor to have species named after you. A few years ago, my colleague Jason Bond got to be on the Colbert Report because he named a species of tarantula after Stephen Colbert, Aposticus Stephen Colberti. So a lot of the times the researchers will derive the names based upon people they respect, people they want to honor. So these are honorific names, species names. But there can be fun species names as well. So one of the most fun names that I ever helped create was in 2008, my colleague Jack Burkhart and I published a paper in which we described new species of sulfugids, of camel spiders. And the type specimen of one of these species in the genus Hemerotreca was collected at the Nevada test site, which used to be called the Nevada Nuclear Test Site, where we early on tested nuclear explosions. So we named this species Hemerotreca kaboomi after that first nuclear explosion. And then there are other fun names out there. These are just a few of them. There's a fairy fly that's named after Tinkerbell, Tinkerbella nana. There's a wasp genus Pisan, and Menke named a species Pisan-yu as just kind of a fun pun. And Terry Irwin and Irwin, there was a beetle genus called Agra, and they several years ago named a couple species Aggravation and Agracadabra. There's a species of snail that's named Ba humbugi. So Ba is the genus, humbugi is the specific epithet. There's a genus of wasp called Here's. So Marsh named a couple species, Here's to ya, and Here's looking at ya. There's a species of that is named in honor of SpongeBob SquarePants, because apparently the fungus looked kind of like SpongeBob, so it's called Spongiforma squarepantsii. And then a few years ago, my colleague Norm Platnick, who died last year, unfortunately, but Norm was examining this huge genus of spiders called Knops. And he realized that several species that were dumped in that genus really belonged in their own genus. They were different enough to be placed in a new genus. So he created new genera names that he named not-nops, taint-nops, and tisn't-nops. So I'll leave you with some take-homes. One is, if you were to designate, who do you think we should designate as the Homo sapiens holotype? Your choice of who should be our holotype. If you were describing a new species, who would you honor? Would you create an honorific, and who would that be based upon? Or would you work a pun into the name of a new species? Did you find some of the names today kind of funny? And did you ever realize that taxonomists had a good sense of humor? So I'll leave you with those questions to ponder, and I'll open it back, throw it back to memory to see if we have any questions.
Awesome. Hello again, everyone. My name is Memory Williams, and I work in the marketing department at the museum. Just popping in like I always do to read your questions to our presenter. Today we have Dr. Paula Cushing. What an awesome presentation. That was very enjoyable.
I think it's fun. Some of the names that people create are a lot of fun.
Yeah, I love the SpongeBob one. So if you have any questions, just drop them in our Facebook chat. I'm also looking at YouTube. Just a few comments we've gotten so far. One from Matt, who says, I thought Linnaeus was the designated holotype, referring to something.
So Linnaeus described the species, but he never designated a holotype. So there's no actual specimen that's deposited anywhere that represents the Homo sapiens holotype.
Okay. Oh, Phyllis just added, one of my favorite snail genus is, I'm gonna miss it. Itibitium, very small snails.
Itibitium snail.
How cute, I love it.
Very descriptive.
Yes. And just in case we don't get any more questions right now, feel free to leave them later on as you watch, and I'll be sure to get them to Paula. But what some awesome takeaways too. I was sitting here thinking about what I would think about if I were to name a species and who I'd like to honor. And, that's something that I've always thought about, but I feel like I would be one of the people who would just choose based on looks, like, oh, this looks like this to me, so I'm going to do this.
Well, it's interesting because we had a zoology curator meeting this morning, and we had this interesting discussion where some of us felt that honorifics were good, to honor a person. But my colleague, Gar Spellman, who's in the ornithologist, he felt like in his field, it was considered not so appropriate to name species after people. So there's differences of opinion. Yeah. And one thing I didn't mention is that there are official committees that will decide upon species name disputes that are called, the ICZN committee is the one that we work with, International Code of Zoological Nomenclature, and there's an International Code of Botanical Nomenclature. And in fact, another of my colleagues, Frank Krell, is a commissioner with the ICZN. Really? So Frank is one of those who, if there's a disputed species name, he and his colleagues at the ICZN will make the decision about which way it should go, what name is most appropriate. There are all sorts of rules that you have to adhere to when you're naming a new species. And one of the rules is kind of interesting. There is a rule that says you shouldn't use words that are rude. You shouldn't insult people. So I think a lot of taxonomists have kind of cheated and maybe named, fungus species after those who they didn't really like, or including little insults in species names.
Interesting. Wow, what a cool job. Someone who decides what to name a species when there's a dispute. I mean, who knew? Okay, we have a few more questions coming in. One from Phyllis, who asks, what's the best way to search for details about the holotype?
So the best way is to go, I showed that picture of that really old book. So the best way to search for information about holotypes is you look at that. Remember I said that there was a name of the describer that is usually listed after the species name. So you can use Google Scholar or some online resource to try to track down the original paper in which that researcher described it. It might be all the way back to 1757 to Linnaeus, or the one that I showed you was Clerc, who's described a lot of spiders back in 1759. And so it might be going all the way back to those original resources and looking at the information about the holotype. But that's kind of what you oftentimes have to do, is go to the original paper where the species was originally described. It's a great question.
Yeah. We have another one from Sarah who asks, is it possible to pay taxonomists to name organisms specific names?
That's great. That is a great
taxonomist.
Well, that's a great question. And actually a few years ago, so that's kind of controversial, because some taxonomists have used species naming rights as a way to kind of raise money for particular causes. And there's a controversy in the field of taxonomy about whether or not that's a good idea. A lot of taxonomists feel like money should not be involved. That really creates a can of worms that you don't want open. And others feel like, well, if it's for a good cause, if it's to support the collection, if it's to support some, climate initiative that's saving a particular habitat, maybe it's worth it. So it's sometimes done. Money is sometimes involved in species naming, but I think the trend is that maybe is not such a good idea.
Yeah, that makes sense. And then, if I were super rich and just wanted to name a lot of things after me, I don't know who would want to do that, but you never know.
Well, and that reminds me that it's also considered very, very unacceptable to name a species after yourself.
Yes, I remember that. Okay, we have another question from Brandon on YouTube who asks, why is it so difficult to find new species? Have we found most of the large ones looking for smaller and smaller species? That's a great question.
That is a great question. And it's not so much that it's hard to find or recognize new species, it's just a lot of work. And so if you think about these museum collections, particularly the ginormous museum collections like you have at the American Museum or the Smithsonian, I am 100% certain there are new species sitting in those drawers waiting to be described. But it requires researchers who know enough about the group to go in and to recognize that they're seeing something new. And so the problem is really not the discovery part of it, but it's the description part of it and the work that is involved in describing the new species and recognizing the new species, because there aren't enough taxonomists. So the field of taxonomy and systematics, there's a lot of ecologists out there, there's a lot of animal behavior people out there, but they don't have the tool set to recognize a new species. The number of taxonomists and systematists, there's just not enough of us out there to do the work that's involved in describing the species. But that's a great question. I think it's a lack of scientists rather than a lack of species that are sitting in drawers or sitting out there in your backyard that are waiting to be discovered.
Interesting. Just looking for any other questions. Yeah, I was kind of thinking about that too. I was wondering are there a lot of opportunities to name new species? Are we finding new species every day, or I don't know the rate, but is there always an opportunity to name something new or to describe something new?
If you have the knowledge for that particular group to recognize it, then the opportunities are definitely there. So even in my arachnology collection, I'm 100% certain that there are species that are waiting for me to go in and pull them out and say, oh yeah, no, this is new.
Interesting. How about we end with this comment from James, who says, I like descriptive names, but did name an ant for E.O. Wilson, only I called it Formica biophilica.
Wonderful.
Of his beautiful concept, biophilia. He wrote me a nice postcard thanking me for such a thoughtful name. That's nice.
That's fantastic. Yeah, that's a great name.
Yes.
And then what a great way to honor such an incredible scientist as E.O. Wilson.
Yeah. Oh, how about, actually, I just saw one more. question come in. We'll end on this question from Matt. So what is the current thinking on naming species without a physical holotype, using a video or blood sample and releasing the specimen?
That's also a really good question, and that is also controversial. So for those, some of us who, especially those of us who work with arthropods, there are such subtleties involved in recognizing that it's a new species that we strongly feel, I think in general, that it's good to have that physical specimen. But there are other scientists who feel like DNA technology has advanced to the place where a blood sample and good photographs from the field are sufficient documentation of that new species to name it without having the physical specimen. So to a certain extent, it depends on whether you're working, what field you're working on, whether you're working in vertebrates or with plants or with arthropods or with bacteria. And then there's a philosophical and an ethical questions that go into that. So that's very controversial. Some of us feel like you really need that type because that is a physical manifestation and a physical representation of that new species, and you can't have anything better than that. Yeah. But others feel like maybe we don't need that anymore. Great question.
Interesting. Yeah, that's a great question to end on too. So I'm definitely going to go into my weekend thinking about what I may name a species I discover. Thank you so much, Paula. Anything else you'd like to leave us with as we get on out of here?
No, just to recognize how much diversity is left to discover on this one planet. That's why what we do is so important, is we need to know what we share our planet, our home with.
Awesome. And we will be right back here next Thursday. Next Thursday is Earth Day, so we will have a special Science Division Live for Earth Day. But until then, thank you so much, Paula. Thank you all for watching. We'll see you next week.